So thanks everyone for coming during the summer break. It's a great turnout for this time of year. It's my great pleasure to introduce Mr. hopefully soon to be Dr. Guangqiao Sun. He's been in our lab since 2013, working on his dissertation he's going to present today. Prior to that, he was an undergrad at Northwest Agricultural and Forestry University in China. He did a Bachelor of Agronomy with a major in plant protection. He was there from 2009 to 2013. He did two undergraduate projects while he was there, one evaluating and analyzing the tDNA insertion mutant bank of apple Valsa canker pathogen Valsa mali, and investigating the national distribution of leafhopper species in China. So he's been very active in doing research. And while he was an undergrad in China, he was part of a group of about 60 undergrads, I think. There was a big group came from Northwest University to UNL and engaged in some undergraduate research opportunities through the summer of 2012, I believe. I think it was 2012. No, but 2012 was the year, I think.
2012.
Yeah. And Sun came to my lab for about 6 weeks to work on some things that kind of developed into this story later. He went back to China, liked UNL so much, he applied for a Chinese Scholarship Council fellowship and was awarded that, and that funded him to come here and do his PhD in my lab. So since being at UNL, he's won a number of awards, such as the Widerman and the Larrick Award, and he also won an award for his poster at last year's UNL Plant Science Retreat, which was pretty cool. He's presented his poster at a couple of national and international meetings, including the this year's Fungal Genetics Conference, which is a big international conference held in California. He had a poster there, but he also got up and asked a question in the main session in front of 2,000 people, and the speaker only answered half of that question. He didn't want to say anything at all, so he kind of Sun got him on that one. So I think that's all I want to say, and with that, I'm just checking there's nothing else. I'm going to hand it over to Sun.
All right, thanks, Dr. Wilson. Right here, and thank you everybody for coming here. I know it's a time to finish things up doing this, but it's also time to start things all over again. So I really appreciate your participation in my thesis defending. So my PhD projects is I'm trying to answer the question that how pathogen cells feed on their host cells. How does a pathogen like Magnaporthe oryzae manipulate the metabolic strategies to facilitate the invasive growth in rice plants? So to answer this question, I have 3 chapters in my thesis, from chapter 2 to chapter 4, representing my research projects. So these are the chapters I'm going to present today. But before I get into my projects, let me show you the significance of our work, or the significance of doing research on rice blast disease. First of all, I believe that some of you had rice for breakfast, and some of you are going to have that for lunch. So rice is consumed and produced all over the world. If you look at here, the distribution here, in Southeast Asia, where we have 50% of the global population living here, and is the center of rice production. Remember that we have half of the population, global population, living here. So rice is feeding the global population. As the population of the world is growing continuously, there is an urgent demand of increasing the rice production every year. So, but this work is really hard. We only have 1.3% increment in rice production last year compared to the year before. So why is it so hard? Because the center of rice production is also the center of Magnaporthe region, diversion, and dispersion, right? So in this area, we lost the most rice to the disease past, right? So that's why it's really hard to get rice production increased. So right now, rice blast fungus still imposes a very huge threat to these developing countries where we have most of the population living in. So how does it look like? on rice plants. These necrotic lesions on leaves is usually the most typical symptom of rice blast disease in the rice field. But this fungus can find its niche all over the rice plants, even from the root. I think this is one of the reasons why this pathogen is always listed on the top of the top 10 fungal, devastating fungal pathogens. It's also considered as a biological weapon by the military. So think about a missile contain spores of Magnaporthe and then boom, in the rice field, right? So how does it infect? The infection always starts from the landing of the three-celled chlamydia. Once it's landed on the rice surface, it can recognize that hydrophobic surface. And then during a due time, it can germinate and form an essential infectious structure called an afrosorium. The afrosorial cell is highly myelinized in the cell wall, which makes it very rigid because inside of this cell, due to the accumulation of glycerol, it will generate a huge physical turgid pressure which will act on this thin penetration pack, which will eventually rupture the cuticle of rice surface. Once inside, you will undergo 4 to 5 days biotrophic growth. All right, later on, it switched to a necrotrophic growth, and they eventually cause these lesions. And this is also the place where the chlamydia are produced. So I would say that the cell cycle, the infection cycle of this fungal pathogen is nutrient sensitive. I guess all the infection cycle of all the pathogens are nutrient sensitive because the ultimate goal for the pathogens is to find the nutrient, to feed, to grow, and then propagate, right? So how does Magnaporthe sensitive to respond to nutrient conditions? First of all, during the afrosorium formation, this stage is nutrient free because there's no nutrient out there, right? So once inside, it's nutrient rich because they are inside the rice cells. So there are a lot of nutrients here. You need to be aware of those conditions, right? First of all, here, once inside, they find the nutrients is good. It's important to know there's nutrient right now. I can grow, but it's also important to sense the nutrient-free condition because once the condition change, say here we have nutrient-rich condition or the fungus is not aware of the nutrient starvation, then the afrosorium will be abolished, right? So no afrosorium, no infection. In turn, it will undergo a vegetative growth, which makes no sense to Magnaporthe because if you undergo this kind of growth, then you won't infect the rice plant, right? So now the question is, what is the regulator that controls the cell growth in response to the nutrient conditions, right? So this is the question I'm asking, and also this is the topic that I think this is the topic of my project, my PhD projects, target of rapamycin, which has been widely recognized as a central regulator of cell growth in response to nutrient availability and quality. So when TOR can be activated by good nutrient, in turn it will promote cell growth. But in turn, it can be inactivated by rapamycin or poor nutrients. So this way, TOR will inhibit cell growth and also promote autophagy. Right? Remember, when TOR is active, autophagy is suppressed. But when TOR is inactivated, autophagy will be promoted. All right. So we know that from our previous study, TOR plays a very important role in controlling the pathogenicity of Magnaporthe. First of all, during the afrosorium formation, from the germinating spores to afrosoria, TOR has to be off. This TOR has to be inactivated. However, if TOR is on, for example, in this ASP4 mutant, ASP4 is the GATA family transcription factor, which controls glutaminolysis. If you knock out this gene, the glutamine level will be elevated and the TOR will be activated. So TOR is always active in this mutant, so you abolish for afrosorium formation. Next, once inside, TOR has to be on in order to initiate the biotrophic growth. So TOR has to be on in response to glucose metabolism. However, in this mutant, TKL1, transcyclase is an enzyme that converts glucose to ATP, which will activate TOR through pentose phosphate pathway. If we knock out this gene, then glucose cannot be converted into ATP, so TOR is off in this stage, so autotrophic growth is not initiated. Given the importance of TOR, we want to know more about it, right? So right now, I know that I know that TOR controls adversarial. Actually, it determines adversarial information in response to glutamine, which is the most important nitrogen source. But I'm asking, how about glucose, which is the most important carbon source for cell growth, right? So this is my chapter 2. We demonstrated that TOR modulates cell cycle in response to glucose limitation to control adversarial information. This is achieved by functional characterization of a glucose-responsive gene, ABL1. So we found this gene by a transcriptome analysis. Right? We found that the expression of this gene is greatly increased in response to glucose, but the expression of this gene is abolished in TPS1 mutant. TPS1 is a glucose sensor that directly responds to glucose 6-phosphate. Right? And when we knock out ABL1 in TPS1, also we don't see anything different. So that indicates ABL1 is downstream of TPS1, which we also put ABL1 specifically to the glucose sensing pathway. All right, so we knock out this gene, the first thing we test is the pathogenicity. We found that it lost its pathogenicity, so why does it lose its pathogenicity? Usually the first thing we check is our adversarial information, right? You can see here that by 24 hours on hydrophobic surface, ABL1 always form these 4 distinct morphotypes. All right, and the adversarial information is greatly reduced. It's around like 35%. All right, so we're asking that, are these adversarial formed by ABL1 functional? So we tested the adversarial information, adversarial on leaf sheath. So we inoculated the spores to leaf sheath and examine the penetration by the adversarial formed by ABL1. We found that only 20% of those adversarial can penetrate into the plant. Even though they penetrate into, they cannot grow inside. So indicating the adversarial formed by ABL1 are hardly functional, I would say. So it seems like ABL1 is largely required for adversarial information, especially the functional adversarial development, right? So we decided to explore how ABL1 controls adversarial information. If you look at those long germ tubes, right? So it's telling us that longer germ tube is, the more rounds of cell division will be taking place, right? So we hypothesized that there might be more rounds of mitosis going on. So we decided to examine the mitosis in ABL1. First, we utilized this strain wild type, in which histone protein 1 is labeled by RFP. Histone protein 1 is the linker between the chromosomes. So the RFP signal allows us to visualize the position of the nucleus. So you can see that by 24 hours in the wild type, we only see 1 nucleus in the adversarial. So the normal process of adversarial formation is that at the very beginning we have 3 nuclei in the chlamydia, because it's a 3-celled chlamydia, and only this cell, this chlamydia, the germinating chlamydia cell, will undergo 1 round of mitosis. That indicates, and there will eventually after the 1 round of mitosis, we will see 4 nuclei. And the daughter nuclei will migrate into the adversarium, and the mother nuclei will go back. And then, due to the autophagy, all these 3 nuclei will be degraded. All right. So that's why we only see 1 nucleus in the adversarium. However, if you look at ABL1, we see 1, 2, probably 3, 4. Right. So more rounds of mitosis is going on in ABL1, and also we need to notice that here we have 3 nuclei in the chlamydia, indicates there's no autophagy happening in ABL1. Also, if you look at the nuclear counting, as I said, if the 1 round of mitosis has completed in wild type, we will see 4 nuclei in spores, right? So we only see most of the spores finish the first round of mitosis by 8 hours. But in ABL1, it is completed in 4 hours. So indicating the cell cycle is greatly accelerated in this unit. But why? All right, we also we also did MDC staining, which labels all the phytosomes. So we can see that, all right, the distribution of all the phytosomes are misregulated in all the morphotypes formed by ABL1. So we're asking that which pathways is ABL1 working? We have two pathways, CPK pathway, TOR pathway. Right. From the previous work, we know that CPK pathways promoting adversarial formation is a major pathway that promotes adversarial formation. But TOR works downstream of this pathway. So that's why we say TOR needs to be off in order to form adversarium. If TOR is on, it will work downstream of this pathway, block it, right? So we can test this by CNP treatment first to activate the CPK pathway. We found that no adversarium is restored. Second, as a rapamycin to inactivate TOR, we found that, okay, adversarium is restored. So in this case, we are inhibiting TOR, and we got adversarium, indicating probably in ABL1 mutant, TOR is overactive, right? So to test this hypothesis, we also did rapamycin and CNP treatment on hydrophilic surface, which is non-inducible surface. All right. So on ABL1 only responds to rapamycin, and also on glucose, with no glucose growth condition, that RS2, RS3, which are all the TOR readout genes. When TOR is active, it will promote the ribosomal gene expression. So, but without glucose, TOR will be inactive, so it should be downregulated. So in wild type, you can see that RS2, RS3 are all downregulated, but in ABL1, it's still expressed in a high level, indicating in ABL1, TOR is active. So the way that we can downregulate TOR is we cannot delete TOR because TOR is lethal. Deletion of TOR is lethal. So the way we did it is to knock out ABL1 in RBP35 background, in which TOR is downregulated. The story of RBP35 is pretty long, so I'm not going to introduce much about that. You just need to know that RB35 is an RNA binding protein. When we knock out this gene, TOR will be downregulated. So we genetically downregulate TOR in ABL1, we restore adversarium. Finally, we conclude that ABL1 is a novel TOR inhibitor. All right, so right now we want to know that does ABL1 TOR signal act on cell cycle, because mitosis is misregulated in this mutant, right? So we want to know how cell cycle or how ABL1 TOR signal acts on cell cycle to regulate adversarial inflammation. So we know that in yeast, nitrogen starvation or rapamycin treatment will arrest cell cycle at either G1 or G2. All right, so remember, nitrogen starvation or rapamycin treatment are all indicating TOR inactivation. Indicating TOR inactivation will arrest cell cycle at G1 or G2. We also find that only when rapamycin was added before the first 2 rounds of mitosis could we restore adversarium in ABL1, indicating that rapamycin treatment is cell cycle dependent. So now let's figure out which stage is required, which arrest is required for adversarial inflammation. We first arrest cell cycle at G1 or S phase by cyclohexamide or hydroxyurea, respectively. We found that neither of these treatments restored adversarium, indicating that G1 or S phase arrests are not sufficient to induce adversarial inflammation. All right, indicates that during the adversarial inflammation, the cell cycle must proceed through the G1 or S phase. So is G2 arrest required? The answer is yes. If we arrest cell cycle at G2 by bendamyl treatment, all right, so we induced adversarium in both wild type and ABL1. Remember that in ABL1, the cell cycle is accelerated, right? So actually, 4 hour post inoculation bendamyl treatment is actually inhibiting the first G2, but in this case, it's inhibiting the second G2 because the first round of mitosis is already completed by 4 hours in ABL1. So indicating as long as we arrest the G2 in the first 2 rounds of mitosis, we can restore adversarium. So here comes the question that, life has to go on. you cannot arrest the cell cycle at G2 forever, right? So there must be a signal kicked in to push the cell cycle, to progress from G2 arrest. So which signal is this? Our answer is autophagy. So because when you inhibit autophagy by 3-MA, which inhibits autophagism production, so we see that by 24 hours, we see 1, 2, 3 nuclei, right? So as I said, remember that we should see 4 nuclei here. The mother nuclei should return to this conidial cell. But why? Where's the mother nucleus? Actually, it's the mother nucleus and daughter nucleus. They migrate into the aphrosorium together. Right? They are not separated, indicating autophagy is required to separate the mother nuclei with the daughter nuclei. Okay. So now we figured out G2 arrest is required for aphrosorium induction. Second, autophagy is required to push the cell cycle to go through. Next question is, so what ABL1 TOR signal is responding to? Right? We say the ABL1 is a glucose responsive gene. So does this signal respond to glucose? And the answer is yes, because when we add glucose to wild type, we see the apherosoria are abolished and multiple rounds of mitosis, and then we see the morphotypes and also more rounds of septations. All right, so indicating ABL1 TOR signal is responding to the glucose starvation signal. Right, once we have glucose, all the checkpoints will be abolished. So we also figured that glucose signal kicked in before the G2 arrest because benamyl can also arrest cell cycle at G2 and use aphrosorium in presence of glucose, indicating the G2 arrest is downstream of glucose signal. This makes sense because first, the germinating spores are sensing the glucose starvation to induce the G2 arrest, right? So if you add glucose after the checkpoints, you won't abolish the aphrosorium. Makes sense. And the last question is, why when the daughter nuclei migrate into an apherosoria, then it stopped mitosis? Why it stopped cell division, right? So the cell cycle is arrested in some stage. So we are asking that which stage are the nuclei arrested in. So first, if we look at the aphrosorium at 18 hours post-inoculation, we only see 1 nucleus in the apherosoria. And looking at that at 48 hours, it's still there. But if we add glucose at this time point and look at the apherosoria at 48 hours post-inoculation, what we see is, we initiated the mitosis, and we have more rounds of mitosis, going on from the apherosoria cell. However, glucose treatment indicating glucose reinitiates the apherosoria, the mitosis, right? So rapamycin treatment can override the glucose treatment, all right, indicating in this case glucose is activating TOR and then push the cell cycle to start all over again. But if you inactivate TOR by rapamycin, which means we can arrest cell cycle at G1 and G2 in presence of glucose, so we can stop the mitosis. But we want to know that which stage are these nuclei in. So it can be easily resolved by hydroxyurea because it inhibits S phase, right? So if you push the cell cycle go through, if we don't see mitosis, that means the cell cycle must be arrested before S phase. So it got to be at G1 or G0 phase, right? So lastly, we want to know that what ABL1 is exactly sensing, is exactly responding to inside the cells. And we found that it's glucose 6-phosphate because when we treated the spores with 2-deoxyglucose, which will cause accumulation of glucose 6-phosphate, we generated these long-term tubes. So indicating ABL1 is sensing the glucose or glucose 6-phosphate level to inactivate TOR or activate TOR, which when TOR is inactive, that will promote G1, G2 arrest and autophagy and apherosoria. All right. So to summarize the whole story, we know that during the apherosoria formation, during the 1 round of mitosis, we have 2 checkpoints. The first checkpoint is G2M arrest. All right, this is controlled by ABL1 TOR signaling. but this can be abolished by glucose. The second checkpoint is once the daughter nuclei migrate into the epithelium, it goes to a G2 G0, a G1 G0 arrest, and this is also controlled by ABL1 TOR signaling, and which can also be abolished by glucose. So in wild type treated with glucose or in ABL1 mutant, all these two checkpoints are abolished. So the cell cycle is going all the way around, right? So after the ABL1 work, I feel like we are not done yet because there are some puzzles. First of all, what is going on after the G2 arrest, right? So because we don't believe that G2 arrest itself induced epithelium, it's something that G2 arrest is just providing a platform, providing a timing for the epithelium to be induced. So there must be some signals kicked in. The second, actually, if you have a sharp eye here, you can see, you might notice that fundamental treatment to ABL1 on hydrophobic surface restored both autophagy and epithelium. Notice autophagy. We say that TOR is active in ABL1, right? And when TOR is active, it is supposed to suppress autophagy. But why when we arrest G2, we also restored autophagy? There must be another pathway kicked in to promote autophagy independent to TOR. So those are my questions. So that's why I did another project as a follow-up paper, as a follow-up project as ABL1. So this is my chapter 3. Because I don't want to keep you guys for 5 hours in here, so I will just summarize the whole story. First of all, we found that a hydrophilic surface where we have TOR active and CPK pathway inactive, rapamycin and CAMP, which inactivates TOR, activates CPK pathway, can restore epithelial inflammation. But in ABL1, we cannot. Only rapamycin works, but CAMP did not work, indicating that this makes sense, right? We already introduced why, because TOR is downstream blocking the CPK pathway. Very interesting thing is we only see morphotype 1 on hydrophilic surface in both strains, right? Where are the other morphotypes? So this is very interesting to me because you can see that on hydrophilic surface, CAMP, even though CAMP did not restore epithelium in ABL1, but it does restore morphotypes, right? Especially morphotype 3. If you don't remember, type 1, type 3. So all these morphotypes was restored by CAMP, indicating CPK pathway is promoting the morphogenesis, all right? So indicating that we know that morphogenesis, morphotypes formed by ABL1 is due to the cell cycle fine-tuning. So this is for the first time that we figured out CPK pathway also acts on cell cycle. Which cell cycle is this working on? So we found that on hydrophilic surface, Benamil could not restore epithelial inflammation. Remember, on hydrophilic surface, TOR is active and CPK pathway is inactive. Indicates these two pathways are all reversed, right? Compared to the hydrophobic surface. So, are we wrong about the ABL1 paper? No. Actually, we need to inactivate TOR by rapamycin. All right. So, indicating that when TOR is inactive, sorry, this arrow is supposed to indicating that TOR off, along with G2 arrest, one of the epithelium induction checkpoint is released from TOR, and it's also upstream of Benamil. The second thing is we want to know, okay, CPKA, does CPKA work in G2 phase, right? So we arrest cell cycle at G2 and add CAMP to activate CPK pathway. We found that we also restored epithelium. This indicates CPKA promotes epithelium through G2 phase. So also this explains probably on hydrophobic surface. When we add Benamil to ABL1, CPKA kicked in to promote the epithelium and autophagy. This is also confirmed by working on the double mutant. When we knock out ABL1 and CPK background, we found that morphotypes are abolished. And also Benamil treatment could not restore epithelium in this double mutant because it abolished these two checkpoints, right? So we conclude that CAMP and TOR controls two distinct checkpoints in G2 delay, and the bendamyl is just in between these two checkpoints. And also, let's explain why the autophagy is induced by bendamyl on hydrophobic surface in ABL1. Right here, if on hydrophilic surface, we say that we need to activate CPK pathway or inactivate TOR, right? So now we induce aphrosorium by activating CPK pathway when the cell cycle is arrested in G2, and we then withdraw all the treatments from the solution, right? And I view them at 24-hour post-inoculation. We see that autophagy is abolished, and we only see 1, 2, 3, 1, 2, 3, indicating autophagy is required to, to promote the cell cycle. And here, if we add CAMP back, we promoted autophagy again. So does rapamycin. All right. So indicating that both TOR inactivation and CPK activation after the G2 delay, especially after the aphrosorium morphogenesis, may activate autophagy. So the significance, well, then we are thinking about what's the significance of having two checkpoints. Right? We found that, all right, if we look at the CPK aphrosorium formation on hydrophobic surface at 10-hour post-inoculation, we see that only like less than 20% of them formed aphrosorium. But compared to wild type, we basically see the aphrosorium are formed by 10 hours at the same level as we see at 24-hour post-inoculation, indicating that the aphrosorium formation in CPK mutant is delayed. Right? So this checkpoint is required to promote or accelerate the aphrosorium formation. This makes sense because in that unpredictable environment, the ultimate goal, the priority for Magnaporthe is to form aphrosorium as fast as possible, right? So that's the significance of CPK pathway. And also this pathway, the checkpoint of CPK is conserved in the vegetative growth, and the vegetative germ tubes, but TOR pathway is abolished because CAMP treatment at 10-hour post-inoculation also induced aphrosorium, but not rapamycin. And also it's conserved in G2. Only when we arrest G2 and then activate CPKA could we restore aphrosorium formation. And this is to demonstrate that after 10-hour post-inoculation, indicating after the second round of mitosis, TOR checkpoints is gone, and so rapamycin could not induce aphrosorium, but CAMP could. And this whole process is dependent on CPKA. All right, so this is our final model. Actually, in between we also have autophagy. So lastly, given the importance of TOR being involved in pathogenicity of Magnaporthe, we are so desperate to get more TOR-related mutants to see how TOR functions in the pathogenicity of Magnaporthe. So the first thing I did was to knock out the TOR gene in the Magnaporthe, but I failed because conventionally we say that if you screen 200 transformants and you could not get any knockouts, that means the deletion of this gene is lethal. But I screened around 1,000 transformants, so I think it's good enough to prove TOR is lethal. So alternatively, what I did was to perform a forward genetic screen by agrobacterium transformation. All right. We have the basis of doing this experiment is we know that rapamycin can stop the vegetative growth by wild type because you can see that on CM this picture is not great, but rapamycin stops the vegetative growth. So we can select for the rapamycin resistant transformants from this transformation. The procedure of this transformation is principally is easy. You just co-incubate the Magnaporthe mycelium and the agrobacterium together and trigger the infection by a hormone called acetylsyndrome, and then we put rapamycin in the selection media. We just select for the rapamycin resistant transformants. All right. So the next thing is to identify the tDNA insertions because agrobacterium are inserting the tDNAs randomly into the genome of Magnaporthe. So I think this is the most difficult thing to identify the tDNA insertion. Luckily, I got some of the genes that are very interesting. The first thing, one of them is a gene encoding an integral membrane protein with 5 transmembrane domains. I want to point out the 5 transmembrane domains because at that time I was scared. It's really hard to perform any protein work with a protein, with 5 membrane transfer, with 5 transmembrane domains. But luckily, I got some interesting results. About this T-tail PCR, I'm not going to talk much about this. If you are interested, we can talk later. So that becomes my chapter 4. We figured out that actually TOR controls all autophagy-dependent effector secretion, or the other way, TOR-dependent autophagy is required for effector secretion during the biotrophic growth by Magnaporthe oryzae. This is the first time. This is exciting because nobody knows how effector secretion is controlled. All right. So first thing, we knock out the IMP1 from the genome of Magnaporthe, and they rendered rapamycin resistance. Also, that when we put the spores on hydrophilic surface, and IMP1 spores are not responding to rapamycin. So now we want to know that is IMP1 upstream of TOR or downstream of TOR? It can be upstream, that blocking the rapamycin signal to TOR, so it does not respond to rapamycin, but it can also be downstream. So to figure this out, we tested the RS2, RS3, and ATG8 expression. So when TOR is active, as I said, it will promote the expression of these 2 genes. But in turn, as I said, when TOR is inactive, that will promote autophagy. So that will promote autophagy-related gene ATG8 expression. So we found that in both wild type and IMP1, actually in response to rapamycin, the expression of RS2 and RS3 are all downregulated, indicating the rapamycin is working actually. But only the ATG8, you see ATG8 expression is not elevated in IMP1 as wild type, right? So that's just that IMP1 is downstream of TOR, and is also specific to that autophagy branch. So now to test this hypothesis, we grow IMP1 on this nutrient starvation conditions. We found that when nutrient is really limited, IMP1 grow much less than wild type, even cannot form a colony on water agar. So also during the aperisolum formation, we can see MDC staining labels autophagism. So the autophagism distribution is totally messed up. We also found that during the aperisolum formation, IMP1 localized to the vacuolar-like compartments, and when glucose activates TOR, the vacuoles or these compartments are retained in the kinesia, but when we inactivate TOR again, the migration of those compartments are restored. So this is indicating that IMP1 activity is under TOR control. So now called the IMP1, test the pathogenicity. It failed to produce any lesions on this susceptible red seedling. So we want to know why. First, aperisolum, right? So we found that the sporulation is okay. It's more than enough for us to do any aperisolum analysis. But on hydrophobic surface, the aperisolum is greatly reduced in this mutant, and also rapamycin did not elevate aperisolum formation. However, interestingly, when we put IMP1 spores on the rice surface, it formed aperisolum at a similar level as wild type, and also the penetration is okay. Only when it's inside the plant, the growth of IMP1 is retained, is abolished. You can see that by 44 hours, when wild type and its complementary string has already spread into the adjacent red cells, but IMP1 forms this extremely bulbous hyphae and is retained in the first cell, indicating IMP1 is probably required for the biotrophic growth, right? So we want to know that what IMP1 is doing, right? We need to know the function, right? So the first thing we did was look at the localization of IMP1. We can see that it's on those vacuoles in tape in the mycelia tapes, and also these vacuoles are associated with autophytosomes. You can see the shadows. All right, it seems like those autophytosomes are fusing into the big vacuoles, and you can see how IMP1 localized vacuoles, how active are they, right, in mycelia. So it's doing something. The first thing that we want to test is, are the GFP we looked at, are the GFP we look at IMP1, the real IMP1, because when a protein attached to GFP then localized to a vacuolar membrane, the GFP is always cleaved, or the protein is always cleaved. So we performed the Western blot to see that whether this protein is still intact. So we found, okay, the proteins are there. That means IMP1 on vacuolar membrane is still physiologically functional. And we found that also IMP1 is probably involved in endocytosis, because if we incubate IMP1, if we incubate the mycelia with FM464 for 5 hours, and the IMP1 GFP co-localized with FM464. So I'm going to explain how FM464 works. All right, first, in the wild type, or in both strains, right? So FM464 will be uptaken through endocytosis. So FM4 is in the environment. The cells are uptaking it from the environment by endocytosis. And then FM464 will be contained in that small early endosomes. And these early endosomes will then be docked to the big vacuoles. So at 1 hour post inoculation, you can see that those FM464 actually in the early endosomes are surrounding, outlining these big vacuoles in both strains. So later on, by 5 hours, FM464 will be internalized into the big vacuoles. This is what we see, right? So in wild type, it does that. But look at the IMP1. So those early endosomes carrying FM464 are still staying here, staying outside of the vacuoles, indicating those early endosomes are not fusing into the big vacuoles. So question is IMP1 responsible for the fusion between the vacuoles and other vesicles? And to test this, we want to know that we look at the acidification, because acidification of the vacuoles is necessary for the fusions between the other vesicles. So we can see that quinoline, which is a dye that only accumulates in acidified compartments, and we can see that under nutrient starvation condition, in wild type mycelia, where autophagy is elicited, the compartments, the vacuoles are highly acidified. Also, but in IMP1, you can see that there's no acidification of those vacuoles. But AM treatment, amiodarone chloride, which is an autophagy activator independent to TOR pathway. So it can activate autophagy independent to TOR pathway. So it can override IMP1 deficient to elicit the acidification of the compartments. And also you can see that how active the acidified compartments are in this mycelia undergoing autophagy in wild type, but how lonely it is in IMP1, right? So that gives us a clue that IMP1 is required for the vesicles fusing into the big vacuoles. And if this happened to autophagy, that means IMP1 is required to ensure an intact autophagy flux, because the destination of autophytosomes is the vacuole. Right? Autophytosomes engulf everything from the cytoplasm, and then eventually it needs to fuse into the acidified vacuoles so that all the contents inside the autophytosomes can be degraded. All right, because the enzymes in the vacuoles need an acidified environment to be activated. All right, so now if we look at this, MDC staining, which labels autophytosomes in wild type, FM464 labels vacuoles in wild type, overlaps. But in IMP1, very, very less extensive, right? Indicating IMP1, it is it is responsible for the fusion between all the phytosomes and the big vacuoles. Right now, we know the function of IMP1. So is this what IMP1 doing during the implantar growth? So first, we look at the localization. So now we start investigating the biotrophic growth of Magnaporthe. First, we look at the localization. If you notice that the localization of IMP1 is different from that in the vegetative growth, in the vegetative mycelia, because we not only localized to the vacuoles, but also outlined the invasive hyphae, right? This really motivated me to look at the secretion system deployed by Magnaporthe, because let me introduce you the biotrophic growth by Magnaporthe. Once inside, the Magnaporthe mycelia, we will say invasive hyphae, will keep the integrity of the plant membrane because it's a biotroph. So it is surrounded by this plant membrane derived structure called extra-invasive hyphae membrane, EIHM. Okay, and the space in between the EIHM and the invasive hyphae is called apoplastic area, where the apoplastic effectors are secreted in. All right, and there's another important structure called biotrophic interface complex, BIC, which is responsible for cytoplasmic effector delivery. So all the cytoplasmic effector will first be secreted into BIC, and the BIC will deliver the effectors into the right cells. So that means once invasive hyphae spread into the adjacent right cells, the new emerging invasive hyphae will be surrounded by the EIHM again, and the new BIC will be formed. So that means if we label any of the apoplastic effectors the GFP will outline the invasive hyphae. If you label any of the cytoplasmic effector by M-cherry, the red signal will label the BIC, right? So this is what happened in wild type. If you label PWL2, which is a cytoplasmic effector, that will label the BICs, right, in the newly emerging invasive hyphae in the adjacent red cell. And also BAS4, apoplastic effector, labeled by GFP, outlines the invasive hyphae. All right, this is what happened. Then if you look at that again, how similar they are, right? And if you look at them side by side, how similar they are. And if you look at the early time point, how similar they are. So IMP1 and PWL2, BAS4, right? So this is telling me that probably IMP1 has something to do with the effector secretion by Magnaporthe during the biotrophic growth. This is exciting because nobody knows how it's regulated. So now let's look at how effectors are secreted in wild type and IMP1. In wild type, as we predicted, at 24-hour post-inoculation, early stage, the invasive hyphae are outlined by GFP, BAS4, right? And also PWL2 labels is clear BIC. However, in IMP1, it's totally messed up because we see three phenotypes. First one, outlined by BAS4, but no BICs. Second thing, if we see multiple BICs and outlined by BAS4. Third one, BAS4 are expelled into the red cytoplasm, and we don't see BICs or teeny tiny BIC here. So how about later? At 44-hour post-inoculation, we see that wild type is already spread into the adjacent red cells, surrounded by BAS4 smoothly and nicely, form a new BIC, right? But in IMP1, you can see that in all the infected red cells, the BAS4 was expelled into the red cytoplasm, and the BICs are all gone. So you might ask, is IMP1 even expressing the gene, right? Is IMP1 even expressing the PWL2 gene? So we examined the expression of that PWL2 and the BAS4. Actually, somehow IMP1 is even higher than wild type, indicating it's not a gene expression issue. It's a secretion regulation. So now we're asking that let's compare wild type and IMP1. The only difference is in IMP1, the autophagy flux is disrupted, right? There's not an intact autophagy flux. So we're asking, is autophagy required to maintain the EIHM integrity and the big? So I need to mention here that only when the EIHM is broken could the BAS4 be be expelled into the red cell, because the membrane is already, the integrity is already disrupted. So we're asking whether autophagy is involved in keeping the integrity of the EIHM and big. But there's another question: is our previous study demonstrated that TOR needs to be active to initiate a pleiotropic growth, right? So TOR needs to be on, which means that will suppress autophagy. But here we are hypothesizing autophagy probably is required for pleiotropic growth. That means TOR needs to be inactivated. So are we wrong for the previous studies? So we first checked how about if we inactivate TOR during the implantar growth, right? We add rapamycin to both strains. We find that wild type cannot grow inside. It means the TOR, yes, TOR needs to be on during the early growth implantar, but IMP1 is not responding to our rapamycin because it's rapamycin insensitive. So now we are thinking probably later on, at an early time point, TOR is on, but later on, TOR is turned off, right? Because probably that means even in the first cell, even in the same cell, TOR is dynamic. So to study autophagy, we utilize 3MA, which is an autophagy inhibitor, AM, and autophagy activator. In wild type, you can see that if we inhibit autophagy, we phenocopy IMP1, right? BAS4 it's being expelled into the red cell cytoplasm, and the big distributed, diffused, or we have multiple bigs. An AM treatment didn't do anything to big formation or BAS4 secretion, but it did promote the cell-to-cell movement. You can see that how crazy the invasive hive are. They are just spreading into the adjacent cells from different angles. How about IMP1? If we inhibit autophagy in IMP1, it doesn't do anything because autophagy is impaired in IMP1. But if we activate autophagy independent to TOR by AM, we restore BAS4 secretion, we restore big formation. Right? That indicates TOR at a very early time, pleiotropic growth, TOR needs to be on to initiate the pleiotropic growth. Later on, TOR needs to be off and promote autophagy through IMP1, which will promote cell-to-cell movement, big retention, and EIHM integrity. These two structures will maintain the effector secretion. All right. This is our final model proposed by this work. So lastly, I want to show my thanks to my advisor, Dr. Wilson, who gives me very delightful instructions, directions every time when I'm getting in trouble. And my committee members, Dr. Nixon, Dr. Harris, and Dr. Kalimanchang. And our technician, Janet, who basically proofread everything I have written during my PhD. And my colleagues in our labs, Margarita, Lauren, Raquel, Dan, and Xiaobo. And also I have an undergrad student, Kyle. He's already graduated, but he did a very great work helping me with my project. Special acknowledgement, Christian, who is the microscopist. Everybody knows him, I think. Yeah, he cannot be more picky on my samples. I cannot be more picky on the images. And also, I want to thank the Department of Plant Pathology who provided me this good environment. I really enjoy the time studying, working here, and all the people around. Amazing. Thank you.
I found it really interesting that you did both hydrophobic and hydrophilic surfaces. Is it because leaves are hydrophobic?
Yeah.
Okay. I just have never seen that.
Hydrophobicity of the surfaces matters. Yeah. They are CPK. The reason why I investigate the CPK pathway is that the CAMP is produced upon
the recognition of the hydrophobic surface. So when CNP is produced upon hydrophobic surface, CPK pathway will be activated. So it's downstream of surface sensing. So when we put this spores on the hydrophilic surface, CPK will be abolished because no CNP is produced. That's why CNP can be selected.
I wasn't going to ask any questions till later, but would you tell us what the hydrophobic surface was?
We call hydrophobic surface a inducible hydrophobic plastic cover slip. So it's artificial surface, a plastic one.
Which plastic?
Well, if you
We want
want
to duplicate it. We'll get it correctly. Okay.
yeah, for sure.
Similarly, what's the hydrophilic?
The hydrophilic surface is the glass line. So you can easily tell by putting a water drop on the surfaces. If they stand there, that's hydrophobic. If it's red, then it's hydrophilic.
So my question is very broad. Did this pathogen evolve from a necrotroph or a biotroph? So which is the lifestyle of, it's semi-biotrophic, so is the necrotrophy something that its ancestry did not have, or which, do you have any idea?
Well, from the evolution, I'm not sure. But for Magnaporthe, yes, it does deploy at 4 to 5 days, first 4 to 5 days in planta as a biotroph, and then switch to Magnaporthe. Right now, we really don't know that how this switch is regulated. But from here, we can see that when the nutrient, so the 12 pathway is very dynamic. It's like in the first cell, at the very beginning, nutrient is very, very good, right? So 12 needs to be on, but later on, 12 needs to be off. And when the red cells are depleted, so when nutrients in the red cells are depleted, so the red cell will be killed actually eventually. So you need to have another proteins also or some other nutrient sensing regulators that nutrient sensors that sense that stress or sense that nutrient starvation to switch that whole manner. Yeah, so if you think about evolution, think about it evolutionarily. You got to be some point that there's enough pathway being probably emerged to facilitate to facilitate the growth in that condition. So yeah, but I don't have a clear answer for that.
In a couple previous dissertations, there was discussion of how to take this to the field, and a sugar analog for glucose was mentioned. Did you do any work with that as well?
That's a very good question. So 2-deoxyglucose, if you look at the experiment, I only use 0.5% of deoxyglucose, and that is enough to abolish that result information. A higher concentrated deoxyglucose will just trigger a small germ cube, and then that's it. So auto abolish. So I think deoxyglucose will work, but it depends on the price. Like if deoxyglucose is cheaper, yeah, we can, why not, right?
Deoxyglucose poisonous?
Yeah, that's right.
Yeah, higher.
I think it's I wasn't going to ask this until later, but since this is a follow-up exactly on that. So have you considered anything less poisonous to DOG? I mean, like glucosamine maybe?
Yeah.
So something that mimics glucose, but it's not metabolized whatsoever. The glucosamine is very cheap. It's not that.
Yeah, that's what I think we should try. But we didn't try that, but this was fine. But also, I think it also depends on how easily all these treatments can be uptaken by the fungus. Just like glucose or 2-deoxyglucose or this kind of thing can easily abolish the actual information, indicating they are inside, they are permeable to the fungus. But for example, like glutamine, it's not very well dissolved in water, and also it's not very well obtained by these fungus.
Well, glucosamine is the glucose was
Yeah, yeah. So and that's right. I mean, probably sugar is much easier for them.
And then we'll answer some more questions later. So let's.